we all know, we're fortunate to have a number of partners supporting our efforts to face stomach cancer. One of our key partners is ASCO, the American Society of Clinical Oncology, which is actually the world's largest professional organization dedicated to the mission of conquering cancer. We are absolutely delighted that Dr. Pam Maggott of ASCO is able to join us today. Dr. Maggott received her undergrad degree in cell biology from Simon Fraser University prior to receiving her master's degree in epidemiology and biostatistics and PhD in translational research from George Washington University. Dr. Mangat currently serves as the director of Centra, ASCO's Center for Research and Analytics, and will be educating us this morning about a unique and dynamic program that she has spearheaded that is helping literally thousands of patients with cancer access cutting-edge therapies. Pam, thank you again for joining us this morning.
Well, thank you for that very warm introduction, Dr. Ruig, and thank you for having me. And I should clarify that I am ABD, all but dissertation, so coming soon. So I oversee ASCO's the American Society of Clinical Oncology's clinical research program portfolio, which includes a couple national cancer clinical trials, and one of which is the Targeted Agent and Profiling Utilization Registry Study, or TAPER study for short. And I'm really pleased to be able to talk to you a little bit about this study. Couple disclosures, I don't necessarily have any, but I will share that ASCO does receive grants from the following pharmaceutical companies to conduct the study. And I will be discussing off-label use of these some approved drugs. So it is fairly unique that a professional oncology society is developing and conducting its own clinical trials. So therefore, we aim to be very thoughtful about the types of clinical trials and projects that we develop and conduct. And we have a set of guiding principles to help us do that. And one is that we want to be sure that we're asking and answering clinically meaningful questions for both patients and providers. And we want to engage patients and patient advocates throughout the research process. We have patient advocates that serve on our boards and committees for the different trials that we conduct, and involve them in all parts of the process, protocol development, and other materials. We also want to ensure that we're implementing the very ASCO recommendations that we are communicating about how we should be conducting research. So we do that as well. And we also don't want to step on any toes. We're not interested in conducting a research that is already being addressed by others. We're not looking to compete with the NCI cooperative studies. We're not looking to have registrational trials. And so by way of ASCO and how we engage with very key stakeholders, patient advocates, clinicians, regulatory authorities, industry, et cetera, we get a unique perspective of what are the large challenges that each of these stakeholders are facing, and is there an opportunity for us to identify an unmet need? And if so, then determine whether ASCO is uniquely poised to address that. So that sets us up very nicely for sort of how and why we developed the TAPER study. So I joined ASCO about a decade ago to work with Dr. Richard Shilsky, who was the chief medical officer at the time. He's retired a couple of years ago officially in that role, but still maintains principal investigator of the TAPER study. And there were a number of real world problems in practice that were occurring at that time and still occur. And that is patients with advanced cancer who had no standard treatment options remaining, or perhaps none to begin with. If their oncologist recommended, or if they knew that potentially having a genomic profiling test performed could be helpful to identify a potentially actionable genomic alteration. Then there was still the problem of how to obtain access to that drug, particularly if it's FDA approved but outside of its approved use. So they're targeted therapies, already FDA approved, but being used outside of its approved use. And how to learn from that treatment large scale. This was happening in practice. all over the country and still is. And so we wanted to provide a mechanism to be able to access the treatment, as well as learn from that experience. So what is the TAPER study? The TAPER study is a large, pragmatic, non-randomized precision medicine basket study. And our goal really is to identify potential signals of activity of these FDA-approved targeted therapies for patients who happen to have a potentially actionable genomic alteration identified in their tumor, and see if we can describe the efficacy and toxicity of that. Who's eligible for TAPER? We were very thoughtful in ensuring that the patients that would be eligible for the TAPER study were reflective of those patients that are seen in practice. So for that reason, we have fairly broad eligibility criteria. We allow for patients who have comorbidities. A lot of the rigorous sort of registrational trials may not. We have broader ranges for organ function, and ECOG performance status of 0 to 2, where some are only 0 to 1. And essentially, patients with any type of solid tumor could be eligible for this study. Now, the genomic profiling tests are not included within the study, because again, this was sort of built around the framework of clinical practice, that this was already occurring. So what we do is we allow for any genomic profiling test that has been performed by any test platform, any company that a physician has ordered for a patient, as long as it comes from a CLIA certified or CAP accredited laboratory. So sort of test agnostic, lab agnostic, and there's no time limit. If the patient had that genomic profiling test performed 2 years ago, they could still use that for screening for the study. We do have some treatment-specific inclusion-exclusion criteria that's related to the specific treatment that is selected, and that is primarily related to safety. And we do have a set of what we refer to as the matching rules in the protocol, which is a set of genomic inclusion and exclusion criteria, which then corresponds to a targeted therapy. So we have quite a few treatments in the study. We have, since we launched in 2016, we have had 25 targeted therapies in the study. And because it's a large platform-based study, we have drugs rolling on and off at any given point. So we started in 2016. We're still continuing to add treatments, and we're still enrolling patients. So we're very much continuing that way. Those treatments there you see in gray with an asterisk are no longer enrolling because they've completed. We're writing up results or various reasons, and those in black text are currently enrolling. And all of the companies provide the treatment to the patients at no cost, as long as they continue to receive some benefit from the treatment per protocol. Now, this slide is not meant to be read or understood. It's really just an opportunity to show you the heterogeneity of the tumor types that we see in the study. And we have, given the number of targets we have, too, it's not a one-to-one. We may have multiple genomic alterations that could be targeted by one of the treatments. So, think about it, 25 treatments since we started, over 40 tumor types, and up to 25 treatments. There's a number of permutations we could see in how we are organizing what we refer to as cohorts. But really quick, just across the x-axis on the bottom there, those are the various types of tumors we've seen in the study. And then every different color in a single bar corresponds to a different genomic alteration that is being targeted within that cancer type. And how we analyze the data is we employ, I'll keep it brief. People usually seem to sometimes fall asleep during the stat part. But essentially, we have a Simon two-stage design, and we enroll 10 patients in one cohort. And we organize the data into these cohorts. And what that means is essentially we're grouping patients' data who have been treated with the same treatment, are targeting the same genomic alteration, and have the same tumor type. And once we reach a cohort of 10 patients, we pause, we take a look at the results, and if there is one or less responder per protocol, and I'll mention what that means, we will close the cohort for futility, and we'll determine that that's a negative signal. However, if there are at least 2 responders in a group of 10, we will continue to enroll up to 28 patients in that cohort. And if there are at least 7 or more responders, we determine that that's a positive signal of activity, and that should warrant further study. And we provide this data back to the oncology community, the industry partners that we're working with on that to help sort of illuminate or highlight some potential areas in which there could be some further development. Okay. We have over 268 clinical trial sites across the country, across 28 states. And since we launched in March of 2016, we have registered or consented to the study for over 4,100 participants. And of those 4,100 participants, we've enrolled over 2,800 of them. So 2,800 have actively received treatment on the study. And I will note that a lot of these sites are community-based sites. We've been very thoughtful in the sites that we have brought on to the study. And so 86% are community-based, only 14% of these sites are academic centers. And TAPER is a pragmatic study. We wanted to ensure that we are seeing the patients that are reflected in practice in this study. So therefore, we employ that broad eligibility criteria that I discussed earlier. We allow for basically any genomic profiling test that has been performed. We have a limited data collection. We're trying to keep the burden low for both patients and for the sites. We are only collecting the information that is necessary for us to meet the study objectives. And we also allow for physician discretion on the drug dosing and modification. Again, most of these drugs are already FDA approved, or they're all FDA approved actually, which is why we have an IND exemption. However, because the safety profiles are well known, we can sort of be a little bit leaner on some of the data we're collecting. We ask that the physicians treat patients in accordance with the prescribing label, and again, can allow for dose modification as needed. And we allow for investigator assessment of response. So typically in the cancer clinical trials, we're seeing RECIST criteria. There's specific measurements that would correspond with a response or not. And I think I missed the study endpoint. The study endpoint is disease control rate. So we're looking at either complete response, partial response, or stable disease of at least 16 weeks duration. And we have secondary outpoints like progression-free survival or overall survival as well. But in any case, we are also including the physician's assessment of response. Are they receiving clinical benefit? There may be a specific measurement and they didn't meet a threshold for a positive response per the calculation. But what's the overall assessment? What would the physician assess that response as? And we collect that information as well. One thing we've been really proud of and is we reflect fairly good diversity in our clinical trial. The top part of the figure there is the TAPER data, and the bottom part of the figure is based on the US cancer population. So you can see there in terms of race distribution, we are doing quite well in the study in terms of being reflective of what is seen in the US cancer population. With Asian, we have about 4% in both our study and what's seen. Hispanic, we have about 6% enrollment. When we started in 2016, we were at 2%. We've made thoughtful decisions in improving that and have been successful. And we are proud as well to say that we have 12% Black participant enrollment. And that is much higher than what is seen in typical cancer clinical trials submitted to the FDA. It's around less than 5%. And age as well. 30% of our patients are between the ages of 65 and 74. So it's older. Okay, so total number of patients on study to date. As I mentioned, we've enrolled over 2,800 participants. And what's really remarkable is that when we look at the patients who have stayed on 6 months or longer on treatment, that's about a quarter. That's 22%. So nearly a quarter of the patients have been on treatment for 6 months or longer. And in the study, patients are on treatment for as long as they're receiving benefit. If they progress or are no longer receiving benefit, they come off study. And then when we look at patients who have stayed on a year or longer, it's 8%. 8%. And that is remarkable when we think about the patient population that we're enrolling here. These are advanced cancer people with advanced cancer who had no treatment options. And I'm reminded of one story, and I have to be quick because I am running out of time. But I'm reminded of one story of a patient who was seen at the Sylvester Cancer Center in Miami. And we don't know the patient's identities. So this story was relayed to me by the investigator there. And she was, her option that was provided to her by her oncologist was hospice at that point. She sought a second opinion. And that opinion came from a physician who was participating in our molecular tumor board. We have a weekly tumor board as well that's available for oncologists to utilize and consult with. Anyway, this oncologist was a participant on that MTB and suggested she have genomic profiling performed and to see if she's a candidate for the TAPER study. She did. She was referred to Sylvester Cancer Center, where Dr. Carmen Calfa is our investigator at that location. And after 2 treatments, the nodule on her neck had fully disappeared. So that's a really encouraging story. Cohorts to date. So we talked a little bit about that really messy slide earlier with all the funky colors. And those are representative essentially of the histology-specific cohorts. We have basically hundreds of cohorts that we've generated. And we can think of that as essentially hundreds of mini phase 2 trials within this single platform study. We have still 97 cohorts enrolling. Some of those have been rolled up into sort of a histology agnostic cohort. And we have started writing these results up and disseminating them. So we have 43 cohorts that we've reported to date. 23 have been negative, 20 have been positive. We continue to disseminate information as quickly as possible. And we do think it's as important to describe and disseminate the negative results as it is the positive. I know we're low on time, so I'm going to skip this slide, but this is just one example of one cohort where you see there we had 2 patients with complete response, 14 partial responders, and 3 with stable disease of at least 16 weeks. And we've presented that, and that's on our website. Any of the results, all of the results from all of the cohorts are available on the TAPER study website, taper.org. We encourage everyone to check that out. And one thing that we're particularly proud of as well is that not only are we working to disseminate the information to the scientific and oncology community, but we're working hard to ensure that we provide information back to the patients and the patients' families that participated in the form of what we refer to as plain language summaries. So, and essentially an easier to understand version of what is the study, what were the results, what came of that participation. We're not sending individual level results. These are aggregate results, but we are taking thoughtful steps to make sure that this is available. And I think this is second to last. I think this is it. We've also been very much engaged, Rich and I, since 2016 in engaging with other researchers around the world who we've heard have similar problems in practice and would like to start up their own TAPER-like studies. So we set up a global data sharing collaboration. We have officially Captured study in Canada and the DRUP study in the Netherlands are actively participating. We've talked with all of these other researchers in these countries for these trials as well. We share our study case report forms, we share our protocol, we share any sort of operational guidance in the efforts to ensure that even if cohorts are not able to complete, that we can pool our data together to make sure that we are making use of every patient who donated their time and space to participate in this trial. And lastly, we're very happy to announce that we're now making the limited data sets of the TAPER data available to researchers, ASCO members, so that they can also help us gain insights beyond the primary endpoint. There's just so much that can be asked and answered with this robust data set. We just can't do it ourselves, and there's no sense in waiting. So we now have this available for request on our website, and encourage everyone to spread the word. All right, thank you.